faithfully delivering value and interpreting great books from around the world. Hello, this is Sally's Book Talk. Today I'm interpreting for you a book called Mendel's Demon, with the subtitle A Brief History of Genes. It's all about genes and the evolution of life. So, the title Mendel's Demon comes from Gregor Mendel, the father of modern genetics. Mendel was an Austrian priest in the 19th century and a famous biologist. He spent 8 years doing pea hybridization experiments and came up with the idea of hereditary factors, what we now call genes. Mendel also found the most basic rules for how living things reproduce, opening the door to genetics. To honor Mendel, we use Mendel's demon as a metaphor for the whole set of mechanisms in biological reproduction. These mechanisms are like a majestic figure in a taoist robe. The king directs genes to function normally, driving life on Earth to evolve from simple to complex. But complex life emerging isn't easy. And life on Earth started 4 billion years ago, yet even today, most organisms are simple like bacteria. Why is complex life so hard to appear? How does the demon drive evolution forward? That's the core question this book aims to answer. The author, Mark Ridley, is a professor in the zoology department at Oxford University and also a researcher at both Oxford and Cambridge. Ridley studied under the famous British evolutionary biologist Richard Dawkins, who wrote The Selfish Gene. Dawkins said Mark Ridley is one of today's top evolutionary thinkers. And in this book, he untangles a key issue about evolution. The book has 10 chapters. The author starts from the birth of the simplest life on Earth, explains how genes got more complex over long evolution, and finally, complex life like humans emerged. Next, I'll break down the main content for you into 3 questions. First, why is it super difficult for complex life to emerge? Second, how did life on Earth go from simple to complex? Third, how do humans deal with their own gene replication errors? Let's start with the book's first question. Why is complex life so hard to appear on Earth? It's the result of natural selection. We can look at the evolutionary journey of early life on Earth. In fact, life on Earth appeared 4 billion years ago. But for the first 2 billion years, half of that time, life stayed in its simplest form. The earliest forms of life were DNA and RNA molecules. They emerged 4 billion years ago with quite simple structures. For example, DNA molecules consist of 2 nucleotide strands forming a double helix. And early DNA and RNA were exposed in the natural environment, not contained in cells or viruses like today, until prokaryotic cells appeared and DNA and RNA got their coats. Prokaryotic cells are cells without a defined nucleus, with DNA exposed inside the cell. To date, the earliest prokaryotic cell fossils found come from Greenland. They lived about 3.7 billion years ago and are somewhat similar in structure to modern bacteria. We call them archaea. Archaea not only have a simple structure, but also carry very few genes, only about 2,500, whereas modern mammals generally have tens of thousands of genes. After archaea appeared for nearly 2 billion years, life on Earth didn't become more complex. Not even a fully formed nucleus evolved. Why is that? The first reason is that simple life can replicate and reproduce more rapidly. American scientist Sol Spiegelman once conducted an experiment simulating early life evolution. He selected some RNA molecules about 3,000 nucleotides long and cultured them in test tubes. RNA molecules function similarly to DNA but are simpler. Some viruses have RNA as their primary genetic material. No, theoretically, in a test tube environment, RNA length could evolve in different directions, some longer, some shorter. But in reality, no matter how long the experiment ran, no longer RNA appeared. Almost all RNA molecules became shorter, with the shortest being about 550 nucleotides, which is the minimum length for RNA to exist. This is because simple RNA can replicate and reproduce quickly, adapting better to the environment, and gaining an absolute advantage in competition with complex RNA. Whereas complex RNA replicates slowly, and in the test tube environment ultimately didn't survive, Spiegelman called this phenomenon the survival of the skinny in Earth's early days, where these skinny beings without extra baggage were better adapted to the environment, but in so at mass siwis liquid. Moreover, these simple, skinny beings are less prone to errors in gene replication, which is why simple life is better adapted to the environment. The second reason, and it's the key point the author emphasizes in this book, when DNA or RNA tries to become longer and more complex in function, the problem of gene replication errors can cause them to quickly make mistakes and die. There are several types of gene replication errors, including mistakes, omissions, duplications, and so on. It's like asking a scribe to copy the word Himalaya. They might copy it as Himalala or Himalaya Yaya. Of course, not all these errors are bad. Take duplication as an example. For instance, suppose you're passing on a message. There's a tiger, run fast. But you copy run fast 3 times, so it becomes, there's a tiger, run fast, run fast, run fast. For the listener, the content of the message is increased and the warning is stronger. Similar gene replication errors can not only increase genetic complexity, but also be very beneficial to organisms. For example, a certain herbivore might have a gene replication error that causes thicker tendons in its legs, allowing it to run faster, which is more advantageous for its survival. But the vast majority of gene replication errors are very harmful and can become genetic defects in organisms. For example, copying there's a tiger run fast as there's a mouse run fast, or there's a tiger don't run. Such gene replication errors make up the majority and are not beneficial to organisms. Taking humans as an example, serious gene replication errors can cause congenital deformities. Congenital diseases like deafness, muteness, congenital dementia, and some cancers are also caused by gene replication errors. In human society, these individuals can receive good medical care and protection, but in the natural environment, organisms with gene replication errors struggle to adapt and are easily eliminated. Moreover, the more genes there are, the easier it is to make errors during replication. Just like a scribe with more tasks is more likely to make mistakes. And the DNA of the oldest archaea on Earth typically has only about 2,500 genes, whereas in structurally complex human DNA, there are about 60,000 genes, so the number differs by several dozen times. The most crucial issue is that every time a cell divides, the entire DNA must be replicated. Errors can accumulate over time. Suppose there are 2 organisms, A and B. B has twice the number of genes as A. Each time B replicates, the probability of an error is twice that of A. So after 10 replications, the error probability differs by 2 to the 10th power, which is nearly 1,000 times. Therefore, it doesn't take many replications for longer DNA to accumulate more errors, eventually becoming unrecognizable. Above, we've covered 2 reasons why complex life struggles to emerge. First, simple life reproduces more rapidly and adapts to the environment more easily. Second, as organisms become more complex, the probability of gene replication errors increases dramatically, leading to elimination by natural selection. That's why Earth's life remained in its simplest state for the first 2 billion years. So how did life overcome the gene replication error problem in the next 2 billion years to evolve from simple to complex? That brings us to our second question. For life to become complex, it must keep gene replication errors within manageable limits. Scientists calculate that organisms must average fewer than 1 faulty gene per offspring to ensure species survival. During their journey toward complexity, organisms gradually evolved 3 main mechanisms to prevent gene replication errors. These mechanisms are like magic spells cast by Mendel's demon, helping organisms evolve toward higher complexity. These 3 key mechanisms are gene proofreading and meltdown, sexual reproduction, and gene confusion. Let's start with the first mechanism, gene proofreading and meltdown. Simply put, the gene proofreading mechanism involves 2 main types of enzymes appearing in cells: proofreading enzymes and repair enzymes. Proofreading enzymes check if replicated genes are correct. If not, repair enzymes fix the errors. It's like giving an unreliable scribe both a proofreader to spot mistakes and a repair specialist to correct them. Uh, whoa, uh, yeah. But these enzymes can't catch and fix all gene replication errors. If errors are too numerous or severe, some slip through. That's when cells deploy their last resort, the meltdown mechanism. The term meltdown uses the character for melting with fire radical and destruction. Simply put, the meltdown mechanism tells cells with severe genetic errors, stop replicating and self-destructive. At this point, to protect the species' genetic integrity, the cell heroically sacrifices itself. One falls, so many may thrive. While the meltdown mechanism sounds scary, organisms have used it since ancient times. For example, certain human gene replication errors can cause uncontrolled cell proliferation. And so, these cells are cancer cells, but the human body naturally produces them.
Once the meltdown mechanism detects cancer cells, it activates their self-destruction protocol, forcing them to self-destroy immediately.
Through this mechanism, the body spontaneously eliminates most cancer cells, preventing fatal cancers. This marks the gradual refinement of gene proofreading and meltdown. PHDL. This maturation was a key internal factor in Earth's first major evolutionary leap, 2 billion years ago. That's when eukaryotic organisms emerged. Eukaryotic organisms have true nuclei, membrane-bound compartments enclosing DNA. Early examples included algae and fungi. Their DNA was more complex than archaea's, with roughly twice as many genes and correspondingly intricate cellular structures. Though still single-celled, eukaryotes evolved organelles like mitochondria and chloroplasts, with larger cell sizes. Beyond internal genetic refinements, eukaryotes emerged with external help, the Great Oxidation Event. Around 2.6 billion years ago, Earth's environment transformed dramatically. Atmospheric oxygen surged, an event known as the Great Oxidation. While its exact cause remains unknown, oxygen accelerated metabolism and reproduction, fueling evolutionary energy. But further complexity, like multicellularity requiring tenfold more genes, would overwhelm proofreading mechanisms with errors. So, this triggered life's next monumental shift, the emergence of sex. Sexual reproduction became the second key error prevention mechanism. Earth's earliest single-celled life, prokaryotes and eukaryotes, reproduced asexually through direct division. 600 million years ago, sexual reproduction emerged, where gametes fuse into zygotes that develop into new individuals. The author argues sexes evolved specifically to slash gene replication errors. Why can sexual reproduction prevent genetic errors? Because complex organisms may carry minor genetic flaws. Asexual reproduction copies these errors exactly, allowing small mistakes to accumulate into major defects over generations. But in sexual reproduction, while both male and female individuals might each have one faulty gene, their defective genes are likely different, allowing their healthy genes to combine into a complete DNA strand for the next generation. British evolutionary biologist Maynard Smith gave this vivid analogy. Suppose you have two broken cars, one with a dead engine, the other with failed brakes. Both are the same model. What would you do? The smart solution is to combine good parts from both into one working car, turning two useless vehicles into one functional one, which is progress. Of course, offspring could inherit either a perfect DNA strand or both defects. In the latter case, natural selection eliminates them. The meltdown mechanism still activates, sacrificing defective offspring. For example, some animals produce many fertilized eggs. Defective ones degrade to nourish healthy embryos. A species doesn't need all offspring, just enough high-quality genes to maintain population stability. Earlier, we mentioned sex emerged around 600 million years ago. The same period saw single-celled organisms evolve into multicellular life. Sexual reproduction was key to this leap. On Earth, more complex, advanced organisms increasingly favor sexual reproduction. In nature, most vertebrates use sexual reproduction. We observe that stronger, better adapted animals more easily find mates and reproduce. They carry fewer defective genes, reducing transmission risks to offspring. Still, some vertebrates resort to asexual reproduction in crises, like certain sharks and lizards, when populations crash and mates are unavailable, cloning themselves instead. So, but in this case, their offspring are generally very weak. But in sexual reproduction, there's another issue. Not all defective genes trigger the meltdown mechanism for self-sacrifice. Some desperately try to pass themselves to offspring. We call them selfish genes. Mm. On Sally's Book Talk, we covered The Selfish Gene, authored by Richard Dawkins, who is Ridley's mentor. Richard Dawkins. Dawkins argued that some individuals with defective genes ignore species welfare, desperately passing their genes during reproduction. And these rule-breaking genes are termed selfish genes. The most notorious is the assassin gene. During reproduction, it identifies and kills mates' genes, replacing them with its own and offspring.
This transmits defective genes, harming species survival.
To counter selfish genes, life evolved a third mechanism: gene confusion. Simply put, it makes parental genetic identifiers identical, rendering them indistinguishable. Like a Chinese chess set. Originally, with black and red sides, if all pieces are painted same color, assassin genes can't target enemies without risking self-harm. Thus, to avoid friendly fire, assassin genes cease attacks. Through this elegantly simple method, organisms prevent selfish gene destruction. This concludes our second question. To prevent replication errors causing defects, life developed three mechanisms: proofreading, meltdown, sexual reproduction, and gene confusion. They reduced replication errors drastically, enabling evolution from simple to complex. Millions of years ago, Earth's most advanced life, humans, emerged with DNA containing 6 billion nucleotides and 60,000 trait-controlling genes.
Despite vast genes, human mutation rates remain fastest, over double other species, accelerating evolution.
In just a few million years, we evolved from hominids to modern humans, becoming Earth's most successful species. But this comes with risks, like speeding on a highway, arriving faster but taking greater chances. Especially with social progress improving living conditions, natural selection weakens, meaning those with congenital defects, once eliminated naturally, may now reproduce under better medical care. This risks accumulating gene replication errors. So how do humans solve their gene replication errors? That's our third question today: how humans address gene replication errors. Beyond the three methods, proofreading or meltdown, sexual reproduction, and gene confusion, we've evolved two reinforcements. First, natural selection plays a more critical role early in life. From fertilization to embryonic stages, natural selection filters out defective genes. A woman produces 5 to 7 million oocytes in her lifetime, but only 400 to 500 become ova, and just a few get fertilized. Oocytes undergo multiple screenings to become ova. Men produce approximately 100 trillion sperm, with only the strongest selected. Ova are protected by the sticky zona pellucida. Sperm failing to penetrate the zona pellucida get degraded by the ovum. Post-fertilization, only 1 in 5 zygotes becomes a baby, filtering out genetically defective ones. This maintains healthy newborn rates. The second method is marriage. In sexual species, genetically superior individuals are preferred mates. For instance, male frogs with poor genes rarely reproduce.
Female frogs dislike their mating calls. Similar mate selection occurs in humans.
Men and women have become more selective in choosing partners. Those excelling in various aspects are more likely to marry and reproduce. While individuals with genetic defects often struggle to marry and have offspring, of course, human genetic evolution now involves not just natural selection, but also technology. So, how will genetic technology help humans avoid genetic errors in the future, according to the author? The author sees promise in gamete cryopreservation and gene therapy for combating genetic errors. Gamete cryopreservation freezes sperm or eggs for later thawing and reproduction. For healthy individuals, gametes are healthiest during youth, because at initial production, germ cells' DNA best matches parental inheritance. As people age, repeated germ cell replication increases mutation risks. Thus, older parents face higher risks of fetal abnormalities or genetic disorders. And Many young people delay childbearing for careers. Freezing young gametes allows safer reproduction later. This significantly increases healthy birth probabilities. Frozen gametes can even be used posthumously for reproduction. Cryopreservation helps prevent gene replication errors. Gene therapy corrects existing replication errors.
It mimics natural repair enzymes to fix defective genes.
Though still emerging, the Human Genome Project may enable comprehensive defect identification and repair. Now, to the author's opposition, he rejects human reproductive cloning. In 1996, Dolly the cloned sheep was born in the UK, revealing alternative reproduction methods. The author believes that, besides ethical dilemmas, cloning technology is a setback in human genetic evolution, increasing gene replication error rates. Cloning is essentially asexual reproduction, the method organisms used before sex emerged on Earth. It can't overcome genetic mutation issues. For instance, clones might have dozens of mutation errors, becoming weaker than the original. Although gene editing can fix some problems, human genes are too complex to ensure all 60,000 replicate flawlessly. That concludes the third question of the book. For humans, natural selection plays a more crucial role early in life and influences mating, helping filter out defective genes. Among current genetic technologies, the author opposes human cloning as it only increases genetic error risks. He sees gamete cryopreservation and gene therapy helping avoid and eliminate faulty genes, with broad application potential. That wraps up the main content of Mendel's Demon. What does our future hold? Could we evolve more complex functions, like wings to fly like birds or total like longevity? Achieving these requires more complex genes. Is that even possible? Some scientists are pessimistic, thinking human genetic complexity has peaked. More complexity brings unresolvable errors. But many are optimistic that Mendel's demon mechanisms can expand with technology, accelerating evolution for unexpected abilities. All right, that's our interpretation of Mendel's demon. Notes are below the audio. See you next time.